Okay.
All right. Hi, everybody. I assume that everyone can hear me online too. Yeah, okay. Okay, so my name is Grace Edwards, and today I'm going to be talking to you about a collaboration that we have going on with some PIs at Georgetown. So the title of this talk is The Primary Visual Cortex Responds During Spoken Language Comprehension. And the other people collaborating with me are Sophia Lipetsky, Anna Sidel Greenwald, Eli Merriam, Renzo Huber, Ella Streamer-Mitt, and Chris Baker. So let's get started. For those of you who know us at the NIMH, you're probably wondering how we got involved in all of these things. So let's get started with the story of how this collaboration came together. Let's start with some research that was done back in the early 2000s about V1 being activated during speech processing in the blind. So what a lot of people were questioning and asking was, what is the early visual cortex doing when in participants that have been blind since birth? And what this study shows from Burton and colleagues in 2002 is that there is activity for spoken language, language comprehension, which spans not only the language cortex, as you can see here, but also goes into early visual cortical areas, suggesting that V1 is actually being utilized in the blind for other tasks such as speech processing. So these are two different tasks that are being used here. One was having the participants actually listen to words and try and determine what the semantic meaning was behind the words. So for example, bed, rest, awake could describe sleep. And this other task over here, which shows slightly less activity in the early visual cortex, is related to a task to do with the rhyming. So bake, make, rake, which they are asked to find the rhyming word, in this case would be snake. But the take home is that in these early blind participants, there seems to be activity in the early visual cortex associated to speech processing. However, in the late blind, it seems that this activity is significantly reduced. So what a lot of these authors and researchers were suggesting is that there is this gross plastic change of the early visual cortex. So now step in our collaborators, Anna and Ella, and many others. They were under the impression that it is actually quite unlikely that there has been this massive plastic change which has resulted in V1 being used for speech processing, but maybe there is some kind of underlying mechanism that is already available in sighted individuals. So in a study in 2023, Ella and Anna found that when participants were listening to forward sentences in comparison to backward sentences, I will show you the stimuli at some point, there was activity in the language processing regions, as you would expect, more so in the left hemisphere than in the right. And then also this activity along the calcarine sulcus in sighted individuals. So this indeed indicates that this activity doesn't just occur in blind individuals. And if we show this this in a little bit more detail here, you can see the increased activation for speech in comparison to the reverse speech. And this seems to be lateralized to the left hemisphere as well. So you can see this difference between left V1 and right V1, and then a significant difference between left V1 and V2 as well. So it seems to be specific to the early primary visual cortex. So this supports their theory that there is some sort of relationship already evolved that occurs between spoken language and activity that then is fed back to the early visual cortex. And they went on to do a second study within this experiment where they looked at the imaginability of the word. So they wanted to really specify that this is a spoken language effect. So they then also presented subjects with both abstract objects and more concrete objects, and found that this activity remained regardless of the type of object. So even in the abstract cases, the less imaginable words still activated V1. And it seemed to still be a difference between V1 left and right hemisphere, indicating this lateralization too. So this is the point that Ella and Anna came to speak with us. They thought that this was a nice study for having a look at the depth-dependent profile in V1 for language in sighted individuals. And this really piqued our interest because it seems like a nice paradigm. It's all auditory stimuli. It really gives us a good idea of what the feedback profile would be in V1. But I will say that we are still on the fence as to whether this is a language effect or something else, which I'll get into a little bit more detail about too. So in general, everyone was interested in doing the study, but what really this activity is in V1 is still up for grabs, I would say. So the first thing we did was replicate this non-visual activity at 7T. So we took exactly the same design as they had before. So they had a block design, 30 seconds on, 30 seconds off. And the subjects for the forward language were listening to sentences like these. I'm not sure if you're going to hear these online, but you can read them. So you might hear them. Oh, no, hold on. Oh no, that is not going to work. Okay, never mind. So essentially they'd hear sentences like birthday cake lights are candles, or something that sweeps the floor is a canoe. And then their task was to detect if the sentence was semantically incorrect. So they would press a button for that final sentence. And I'm not going to pretend to do the reverse language. You'll have to take it for certain that it's very difficult to understand, but essentially it's the same sentences that we presented for the forward language, but in reverse. And then the subjects were detecting a beep presented at the end of one or two of the sentences in the block. And at the same time, the subjects are fixating on a fixation cross for both the forward and the reverse blocks. That didn't change for the entire run of the 6 runs that we did with our participants. And then the sequence that we used was a whole brain EPI aligned to ACPC 1.8 millimeter with a TR of 1.5. So something that I want to note here is this is where we started to question exactly what that kind of feedback was in V1, because as you can tell, there is a very different task between these two stimuli, as well as the stimuli being different themselves. So in the forward language, the subjects are asked to detect if this sentence is semantically incorrect, whereas in the reverse, they're just detecting a beat. There's an argument that the task difficulty is not totally matched. There's also an argument that one of the tasks may be more arousing than the other, etc. So there are different forms of feedback that could be occurring with this kind of task. But again, I will state that there is no visual information going on here. So whatever we find in 7T has to be some sort of feedback effect. Okay, so this is our result. So this is just the replication with bolds, so large voxel size. So this is not the depth dependent effect, but what we found is that we could replicate their effect for the contrast of forward versus reverse across the whole brain. So you can see this increased activity in the left hemisphere in the language processing regions like Wernicke's area. And what you can also see is activity occurring in left and right hemisphere, the early visual cortex. If we then specify that to V1 and V2, ventral and dorsal, what you can see is there seems to be an increased activity in the left hemisphere over the right, and an increased activity in the dorsal regions over the ventral. So this replicates the effects that were found by Anna and Ella and made us happy to move forward to the next step, which was to move into characterizing the depth-dependent profile of the non-visual activity in V1 using VASO. So I will start by saying, I know that some of you may be getting excited by the idea of seeing a non-visual activity profile in V1, but so far we've only been able to collect one participant for what I will go on to talk about our comparative actual visual stimulus in V1. But I'm going to walk you through exactly what we plan to do, and this is the perfect time to get feedback. So if anyone has any ideas or anything that we should change, this would be very valuable to us right now. So the design of the experiment was exactly what we had already done with just our bold sequence, but now we would be applying a vaso sequence, and this would be our non-visual profile for activity in the early visual cortex. And then with in collaboration with obviously Renzo, we also decided to add this comparative visual profile as well. So this is the data that I will be showing you today. So in the comparative visual profile, we had the participants do a 30-second on-off block design. During the on blocks, the subjects were seeing a flickering checkerboard that was flickering at 60 hertz with a 15% contrast between the lighter gray and the darker gray. And the subjects had to detect a target that was presented for 16.6 milliseconds in amongst the flicker, which was either white or black. It was a fairly easy task, but there were about 6 of these targets to detect per block. So our prediction was...
Good question.
Yes.
That was flickering at 60 hertz?
Yeah.
No.
Well, I mean, it's not right now.
No, but you can actually, does it look like it's flickering?
Yeah. It's like one frame on, it's one frame on and off.
That's really fast.
Yeah, it's fast.
This is on 7TB.
70A.
Okay.
It's like almost gray.
No, it's really, you can really detect it. Every single frame, you can, it's very, I wish I had now a video of it. I do have a video somewhere. I should have put it in here. But yeah, it's very easy to see.
Okay.
Okay, so our prediction was that we would use our comparative visual profile to help us define where along the cortical column we had our central like layer for, if you will. So hopefully with this, which I will show you actual data for in a minute, this would be our comparison to our non-visual profile, helping us determine where our more superficial and deeper layers are. So then with our goal of being able to collect the data of the non-visual stimulus, the language comprehension task, to see if we can find some sort of feedback profile. So this profile that I'm presenting right now is just one of many that we could find. We are not married to any specific feedback profile. So this is the double hump that we have seen in previous studies. But there's lots of work out there to suggest that long range feedback is predominantly going to deeper layers, or short range to superficial layers, et cetera, et cetera. So we don't really know exactly what kind of feedback profile we're going to be looking for. So this is up for debate.
And that would be the difference between the forward versus backward speech. Right?
Yes, exactly. So the non-visual profile is in purple. That is the contrast between forward versus reverse speech.
Which might then be looking similar like the forward versus nothing, would it?
So we never did forward versus nothing. We always do that comparison in order to get the localize the speech comprehension. So it's not just an auditory stimulus necessarily. We do that. So actually we could look at forward and reverse together versus baseline, and then we would just have like an auditory. And then if there's anything in V1, that would just be an auditory feedback to V1. Like, yeah. So the forward versus reverse really localizes it to that speech processing.
But either way, you would expect it to be feedback.
Yes. Yeah. Okay. So our scanning parameters and peripherals, as I said, we were using a vaso sequence with 24 slices, 0.8 millimeter voxel size, TR of 3.802, individual TR of 1.6. We included Nordic at the end of each run for thermal denoising. Kaipi was on, and the slab was aligned to the calcarine sulcus. And we also had an eye link on our participants at all times, so that we're able to record their gaze position density, which looks a little something like this. So this is just one of the subjects that we collected in the bold for the original bold design. And what you can see here is that we could get a good idea of where the subjects are fixating for the forward versus reverse speech. And using that, we can do all sorts of things. It's just one example where we can have a look at the position density to determine if the different tasks also elicit different kinds of eye movements as well. Okay. So this is our preliminary result. We have one subject with just the feed-forward comparative part of the study. So what I'm going to show you here is just the steps through that we took. So the first thing we did was to draw our rim file onto, this is the scaled vaso data. So I just wanted to show so that everyone can see exactly where we draw along the calcarine sulcus. So I'll flick that on a little bit so you can see where we were. Then what we did is we then plotted the t-stat for the bold and vaso in this participant, which is a checkerboard versus baseline. And you can see the minimum and maximum t that we used in order to plot this data. So what is simple to see is that we have a nice activity in the early visual cortex for checkerboards, not super surprising. But you can see that this nicely follows the folding of the cortex in both the bold and the vaso, with vaso being a little bit more specific, but a weaker signal in general. And then if we plot the profile from CSF to white matter, you can see that predominantly the activity is peaking in the center between CSF and white matter, with that shifting over to the right for the vaso data in comparison to bold, which is shifted more towards the CSF. And then as we were discussing, there is that drop in signal for vaso, which is also has been found many times before. So this seems to indicate to us that the subjects are able, that this flickering checkerboard nicely elicits a feed-forward response in the early visual cortex. For those of you that are interested, this is our TSNR for bold and for vaso, which I was excited about. It seems nice and strong. So on average, it's about 120 in the left calcarine for bold and 95 in the left calcarine for vaso.
This is after Nordic.
This is after Nordic. I have some backup slides if people want to see with and without Nordic for all the analysis. But something I will mention is that although the sequence seems to be really nicely designed, we are a little bit susceptible to motion. So this is just the autocorrelation of one of the subjects that we decided not to analyze. And as you can see, there's real structure in this image, indicating that even the slightest motion can result in this strong structure in the autocorrelation. Okay, so things to do. We need to collect the data. The current pipeline, I just wanted to walk through what we have done and what we are planning to do and things that we are starting to think about. So the current pipeline is to reduce the thermal noise using Nordic, motion correction, bold correction, and then we do layerification using ITK snap. And then a simple 3D deconvolve in order to get the checkerboard versus baseline. Our plan is to do layerification across the calcarine using Marco's pipeline that was developed with Renzo in the last year. So currently the data I've shown is just in one slice, so we really have a lot to do. That's going to be a real complication when it comes to implementing this layerification. And then things that Renzo and I started talking about the other day is how we should interleave the feed forward and feedback runs. So at the moment, we are doing separate sessions for those two profiles, but then we obviously are we will run into complications of aligning all of this data. So maybe it would be better to interleave the feed forward and feedback runs within each session. These kinds of things, we thought it might be worth discussing with you all in case you had any thoughts. Yeah, so that is where we are currently. Thank you. I have some backup slides if people are interested in TSNR, how it changes with Nordic, etc. And some things about task as well if you're interested, but I just thought I'd take questions at this point. Thank you.
Hello, Grace. Can I ask you a question about your ROI definition?
Yes, yeah.
So here, how do you define your ROI for those layer analysis? Do you based on the activated region or based on the retinotopy mapping, and do you include both hemisphere? like for
Right.
this?
Yes, so right now what you're seeing is just left V1 along the calcarine. Our goal is actually to take the whole of, so the slices that we have at the moment just sit along the calcarine sulcus, and we will take all of that data along the calcarine and actually do layerification across the whole thing. We don't have any retinotopic mapping, but we could bring in participants that we have mapping for in 3T, and then use that in order to localize better. But because we really are just along the calcarine, we don't have much else than V1. So I guess it's more of just a cutoff along the slab as to where we would stop. But
The slab is kind of parallel.
exactly, the slab is parallel to the calcarine.
So basically based on your like some experience and also based on your activated region, because your activated region is mainly in V1, right, as you said. And do you include the so now you just include the left hemisphere, and in future we will include both hemisphere. So what's
Yes.
okay.
Yes, so the plan is to include, sorry, I should have specified. The plan is to include both hemispheres. There's also the suggestion from our collaborators and previous researchers that there is an increased activity for speech comprehension in left V1 versus right V1. So it would be very interesting to see how that impacts the feedback profile for left versus right separately as well.
Yeah, thank you.
Sure.
Maybe I didn't get the purpose of the eye tracking. What?
Yeah.
What would you need to see to change anything in the
Yeah,
interpretation?
so actually the eye tracking kind of plays a couple of roles actually. So if we see significant differences in how the subjects are holding fixation for those different types of runs, it could have two effects. The first one is that it can actually change the feed forward input too. So there could be some retinal slip associated, which would actually change the activity pattern even in the middle layers. But then also thinking about the feedback effect too. So this is going to change the way that information is being fed back to V1 as well. So it's important to have the data so that we know if there is any significant differences between forward versus reverse. And if there are, at that point, we have to then consider how this is going to impact our activity in more of those middle layers, even in our feedback conditions. So this is something that these previous papers haven't recorded eye tracking data. So we don't know how these tasks are impacting the eye movements in general either.
And from your bow study, that time you didn't.
We did, but we only had one subject that had it was very much in development. So we only had one subject that we collected the eye tracking data on, and that was the one that I showed just there.
Here is a figure from Yu Hui and Peter and myself showing bold amplitude with a 60 hertz flicker.
Yes.
It goes down.
Yes.
You might want to consider a lower flicker frequency.
I can,
Yeah.
but I promise I can show you the stimuli.
Okay.
You can see it.
Yeah.
It's very strong. It's
Right. But
yeah.
that shows a higher response at a low frequency.
Yeah, we can try a low
Yeah.
frequency. That's no problem.
But actually, and related to that, you use low contrast.
because we're trying to lower
The amplitude.
Yes.
Right, so that's a subtlety you didn't
Yeah,
mention.
yeah, I didn't
So
mention that. Yeah.
The point is that the feed forward stimulus should have roughly the same response amplitude as
Right.
the feedback.
Yeah, because
so that
we
you
want
can
to compare
compare.
them. Exactly, which is why we did the 15% contrast between the lighter and darker.
Right, rather than 100%.
Exactly. So we don't want the activity to be super high, but we seem to get something a little bit higher than what you have here. But
Yeah. Right, this figure shows a strong drop off at 60
yeah,
hertz.
yeah.
Right, and the other thing you don't show in the feed forward is the superficial bias. And it's little.
Yeah, it's there. It's little. But yeah.
Usually it's like highest at CSF.
Yeah, but it is possible that maybe my rim file is too far into the CSF. This could be why. But that's why you need kind of both profiles actually. So if you have both
That's right. It looks pretty. Here
yeah, hold on. I can show you.
Only in one part.
Yeah, so Yeah, so this is the thing, this is just one of the slices, but the nice thing about having the comparison between the feed forward and the feedback is that it will actually help us determine where the more superficial and
Right.
deep and middle layers are.
Another thing I've seen Renzo do is plot along the x-axis, the gray matter intensity.
Right,
So you'd wanna
we can do that
see
too.
where there's a drop off, and then that's where you're gonna see a set.
Exactly, yeah, we can do that too.
And then one more comment, I try to agree is important. You could also blindfold subjects, so even if they are moving their eyes, they're doing a feed forward input.
That's true, but we wouldn't know if they were moving their eyes at that point. To be able
Even
to
if they
see.
were, it could be.
Okay, but it could be part of what the feedback would be, so I guess
But
that,
then that would be feedback. That would be
yeah,
fine.
okay,
It's just not feed forward.
yeah, all right.
Then
Okay.
it could be just more.
Soon we'll have electrodes.
That'd be motor activity, but that's feedback.
We can put electrodes on in the
Whenever
7T.
it's- Yeah, we're favorable now, so.
Oh,
Oh,
And if they're blindfolded, you could do
Yes.
EEG.
That's awesome.
okay, let's add the peripherals, exciting.
Yeah, we'll never get this time,
Yeah.
but that's,
Okay, yeah.
or,
that's a nice
or you
idea.
could do it like this out of the scanner with a very good eye tracking. And just see if there are differences in eye.
Right. That's true,
Have you also looked at the blink rate, the data you have right now? Because I could imagine that because people suppress their blinks during task rather than
right,
anything really.
Yeah.
It could be that they suppressed more blinks and meaningful sentences versus
yeah,
less.
How about the beat frequency? So they're pressing the button as many times in the forward and backward,
yes,
because they also, that will affect feelings.
yeah. Yeah,
And then, that's really cool, but so do you think it's specific to spoken language? Or like what if, like instead of you have sentences, you just have a single word like canoe, and then you give them like an actual like figurine or something that they can explore haptically?
yeah.
Do you think you get the same? So is it just imagery, I guess, or?
Yeah, so we don't know. So there is the study that's in the other paper that indicates that the imaginability of the stimuli doesn't necessarily affect, but we can't tell that from this particular study. What we did do, go through all of that, what we did do is we did change the task to see how much of an impact that has. So to try and equalize the task for both the forward language versus the backward language, we just did a beep at the end of the sentence for both forward and backward. And when we did that, we actually found that this dropped out our activity totally. So what we question at that point is maybe the subjects just aren't even engaging with the language at all. They're just waiting for the beep. So this then didn't seem like the right task either. So then the next task we tried was a one back task for the forward sentences and the backward sentences. And what that looks like when you look at the contrast is actually quite nice. 3 of the subjects are showing this increased activity for forward over backward. But then if you look at just the forward activity and just the backward activity, what you can start to see is that what we're interpreting is a contrast between negative bold, essentially, apart from for one participant. So then at that question, the question is, what are we actually looking at? So we tried a few things in the scanner to see whether we could try and equalize the forward versus backward to try and determine whether it really was a language effect or a task effect or what it could be. But to be honest, at this point, we then decided, let's go to the one that actually has feedback, everyone,
Right.
and then we can actually just have a look at that profile, but we can't actually determine that it is a language specific thing. It could be task related too.
Why is there negative bold?
For the one back task?
Yeah.
I don't know. We got it in 3 of our 4 participants and yeah.
So wait, the negative thing is still contrast, so people are kind of more, there's more activity during the scrambled or the
Yeah,
reverse
so sorry,
sentences.
this is the activity profile for just forward and just forward and left and right, and then this is just backward and just backward. And what you can see is that each of our subjects is showing. So I mean, at that point, there could even be no feedback to V1, and we're just interpreting who knows what, because it's a language task that's totally auditory, a one back language task. So I don't know whether, why there is negative bold, but it's not a visual task. So I assume that that's probably why.
You know, it's also, I mean, there's a lot going on in this task, right? And you see responses all over the place. Curious with the layer profile and prefrontal cortex. Like,
Yeah,
why just look at V1?
we don't have those slices. We can't get all the way up to frontal.
Really, even
Yeah.
though you're parallel to the top brain?
Yeah, so you have to still go pretty, like, I mean, in order to make
Yeah.
sure that we're getting like air on both ends of the slab.
It'd be pretty sweet to relate the two.
Sure. It would be, yeah.
Could you pull out the trial by trial in just that deactivation?
Yes, I could.
Because I feel like that would be, so in priming tasks, we see like kind of this decrease over time.
Mhm.
So ultimately, like sometimes the later trials bias your signal considerably more than the earlier trials.
Yeah,
Hmm.
yeah. So, I mean, we would like to, I mean, for this particular thing, because it's bold, we could do that. It is designed as a block design, so it's not ideal. But yeah, we could do that, but we would like to keep the block design for the vaso. So I think we can definitely dig into these things for this data when it comes to actually writing up the paper. But as you can tell, it's going to be a confusing one to write up already.
Yeah.
Block by block instead of trial by trial.
Yeah, we could do block by block.
In fact, you can do block by block contrasted base to rise and base to rise.
Yeah.
Sort of those
Yeah.
things.
That's a nice idea. Yeah.
But also if you are open to modifying your timing, you could do that sort of mixed design where you have like the blocks, but you have a digital trial. Amplitudes that you pull out with like in AFNI stem time.
Yeah, sure.
Because then you could also put in, if they have any kind of reaction, you could
Yeah.
see if you can modulate it that way.
Yeah, that's true. Yeah, we are trying to, I guess we're trying our very best to get the best possible sequence set up to be able to pull out the signal in the vaso, which it's already, just looking at the bold, it's already quite a weak effect. So it's, yeah, it's hard to try and, we want to do so much, but we have to try and focus in on the thing that we're.
How big of an effect is it?
So in just the bold data, we're looking at. Oh. Right. So yeah, signal change of 0.1 in the bold.
0.1.
So it's already very weak. And this is already something we've discussed a lot as to whether, like, what we're going to see in vaso when it comes to the feedback signal.
This is
Because-
percent.
Yes, it's low.
This is the contrast.
Yes.
This is the subtraction of
Yes.
two larger responses.
Yes, so actually I have from just the 3T data, it's already, it's low. I mean, this is just the forward speech, right?
Yeah.
And then this is the reverse speech, and the contrast ends up being much lower, but it's a weak signal in general.
Lorenzo, do you have a rule of thumb? Like detectable?
Yeah, we talked about it. It depends. Like if you want to see a map, like in the feed forward, maybe more than that. If you can collapse the entire patch of the cortex into one layer profile, people have shown consistent results with way less than that. So I
Way
think,
less.
yes. And then kind of
Maybe
at some
try it and
point,
see.
it gets kind of harder to believe it, but people still publish it. And then I think we are in a kind of safe regime here.
Mhm.
If we have the luxury to collapse this entire visual patch, I
Mhm.
think. Yeah.
Princeton, do you have a guideline on like good SFNR? Like if you were to just do like SFNR in sort of like layer patches and like a calculation on like ideal size?
I don't have guidelines. I like my activation maps voxel-wise. There is a study from Irati Makwirigaka, and she said that after 100 voxels, you're out of the thermal noise limited regime, and then averaging more doesn't help you. And that was 100 voxels of open 8. I don't know what patch this is. They're quite small.
Mhm.
Assuming that the entire patch does the same thing, and so on and so on. Yeah.
I think Paul has a question.
Yeah, hi. I was just wondering, do you have to, is this assuming an HRF shape, or are you modeling it with tent or something that allows you to find what the shape is if that's changing?
Yeah, so I haven't actually dug into that at all. It's just assuming that I think it's just the default actually.
Okay, because I wonder if maybe that could have an effect, right? If the shape isn't a very good match for this, then that could really affect your percent signal change that you estimate as well, or especially for contrast and that. So that might be something you could look at.
Okay, that sounds great. Thank you. That's a great idea.
Tina?
Hi, I think you mentioned it, but I think I missed it. How many runs did you get? And I mean, how much
Yeah.
was the scan time?
Yeah, good question. So for the feedback profile, our aim is always 6 runs, but sometimes we only get 5. Actually, we played around with this a little bit. The original study was only 2 runs, and actually that does seem like enough to get like the signal at that point isn't really changing. For the feed forward profile, we also aim to get 6 runs, but we sometimes only get 5. It really depends on the timing of the participants. So the subjects are in the scanner for just over an hour and a half maximum.
Actually, if I could ask another question just about the magnitude. So the regions that you see here, I mean, are these mostly regions where you would expect this signal? So even if it's small, does that give you more confidence in it as well? Because these,
Yes.
my biological knowledge isn't strong, but these look like regions you might expect anyways.
Exactly. So this is Wernicke's area over here, and this is also part of the language network. So this gives us relief that we are activating where we would assume to be activating. However, when it comes to the slices that we will have for our vaso sequence, it's going to be much like we have to rely on this data showing that this is activating the right regions, and we won't be able to have that verification with the slab size that we have.
Okay. And also on a different slide, I think when you were showing the CSF to white matter cross layer curves, there I thought the bold percent signal change was something like 30%. And maybe
Yeah, so this is for the feed forward profile. So that's super strong, yeah.
right. Okay.
Yeah.
Okay, cool. Thanks.
Thank you.
Is that really true? 30%?
Yeah.
Wow.
I'm surprised that 30% fold change is only a t-stat of 6.
Yeah, so 6 is the lower end, right? But the max is 32.39. So
Oh,
that's
nice,
the yeah. So that was my min to max across the whole thing.
I still think 30% at 60 hertz at 12% contrast is surprising.
Yeah. Yeah. It's a very engaging task too. I wonder whether that makes a difference with those like 6 targets that they have to detect. I don't know whether that will make a huge difference. 6 targets per block as well.
I mean, if you took that whole region of interest or whatever, I mean, certainly if you get big vessels, I mean, but
Yeah.
at that small of a voxel size, you can easily get 30%.
Okay. Yeah, so as I said, we haven't really done our real analysis when it comes to the calcarine anyway. I've taken one slice. I mean, this is likely to change a lot when we've actually started to do the layerification across the whole slab.
Grace, so you show a little bit about you talk a little bit about the Nordic, and could you show the TSNR map before and after Nordic to see? It seems it really helps improve a lot of TSNR here.
Yeah, so this is before. No, this is with Nordic, and this is without. Yeah. And if I mean, I can, so what I also show here is like an increase. So I decrease the TSNR so you can see, so the max TSNR here is 200 so that it matches with Nordic. But if I then decrease it, you can see a little bit more of how that impacts the signal. But yeah, it's a big change.
So, like because in your study, your signal change is really low. So, so like for your activation map, if it's no Nordic, basically, do you mean like basically it's not sensitive enough to detect those activation?
Yeah, so this is the profile without Nordic. So you can see, I keep that to the same min and max as shown. So this is what it was with Nordic. Then I keep the min and max exactly the same in this image. And then if I change that so you can see a little bit more of the profile, you can see how big of an impact Nordic has. And this is the layer profile, and this is how it changes.
So it really make a big difference. Yeah. Okay. Thank you.
It's huge, yeah.
I understand that you have like limitation with respect to your slab positioning, but wouldn't it make more sense to see where the source of this activity in V1 comes from? I'm expecting that maybe Wernicke region deep layers might be communicating with deep layers of V1, so maybe a layer-wise connectivity. But I understand the difficulty of doing that, but to me, what I find it quite difficult to understand is that where this activity comes from in V1. That would be for me a bigger question to address than just looking at the laminar profiles of V1. If that's like a possibility, maybe you guys could consider to exploring that avenue.
Yeah, I mean, we can also do a whole brain bold, which we have already. So we can, maybe that's actually where that would be a better place to be able to have a good idea of the connectivity there, and then go into the layers. Then we specify to V1 to try and answer our question. Yeah,
because I think like thinking of a future ahead, when it goes for publication, some of the reviewers may ask for it. And what would be your like speculations about the source of this activity? A visual region responding to auditory stimuli? That's quite interesting.
Yeah. So I do, and I don't know the field particularly well myself. So I do wonder if this kind of thing has already been done. Actually, there's been a lot of work on this speech processing in V1. So I'll have a look and see whether I can find something that already shows this connectivity. But again, I think it's really task dependent. So for our specific task, we would have to run the analysis ourselves to make sure. Yeah. Thank you.
Good luck.
Thank you. Okay.
Thank you. So in 2 weeks, we have the preparation for ISMREM. So anybody who is planning to submit something to ISMREM is invited to give a quick pitch of their projects. See you then.